Hello, everybody. If you are new to our webinar series, welcome, and thank you so much for joining us. Criterion Edge runs multiple informative webinars throughout the year. So if you're interested in more of our webinars, you can always check out our website for some of the ones we've done in the past and to sign up for any of our upcoming ones. For today's webinar topic, we are talking about how to establish the state of the art within the Scientific Validity Report. Our presenter today is Principal Medical Writer with Criterion Edge, Dr. Chavez. Sarah has a bachelor's in biochemistry and a PhD in molecular and cellular biology with 25 years of laboratory bench experience and has taught at the university level for the past 15 years. She has extensive scientific writing experience in an academic environment, as well as for several large textbook publishers. Sarah's strong background in basic research and her extensive experience with regulatory writing allows her to assist companies with IVDR readiness as part of the Criterion Edge team. So with that, Sarah, I'm going to turn it back to you to get started.
Okay, thank you, Jessica, and thank you to everyone who's joining us today. During this session, we will be reviewing the scope and the purpose of the Scientific Validity Report, which I will refer to as the SVR. In particular, we will be discussing how the SVR helps to establish that your IVD is state-of-the-art. So our goal is that by the end of this presentation, you will all have a better understanding of the importance of planning and preparation before you begin to write the SVR, what should be included in the SVR, what we mean by a systematic literature review or an SLR, and then ultimately how to establish the state-of-the-art. So before we begin to discuss how to establish the state-of-the-art, we need to start with a few basic definitions. First, what do we mean by scientific validity? According to IVDR Article 2, the Scientific Validity Report should establish the association of an analyte with a clinical condition or a physiological state. Thus, the basis for each SVR is, in effect, a detailed discussion of the analyte and the method of detection. This is essentially the same purpose of the state-of-the-art section. So IVDR guidance recommends that you should begin to plan how to demonstrate the scientific validity of the analytes as early as possible in your development cycle. However, the SVR is a new requirement, so it's still not 100% clear what the expectations are to establish the scientific validity of an IVD under evaluation. While we all await notified body feedback to clarify the level of detail expected, we must now rely on the extensive experience from the medical device world in order to develop a process and a framework to build on. So how do you go about showing this association between an analyte and a medical condition or physiological state? The SVR will allow you to explain this link by first explaining the background of the clinical condition, the analyte, and the alternatives for diagnosing this condition. Are there multiple types of tests or technologies? What are the pros and cons of each type of IVD? These are all critical components within a typical SOA section of the PER. Next, you will include current recommendations and guidelines related to the clinical diagnostic test for the medical condition or clinical state. This may also include SOA articles, but could require additional ad hoc searches to identify statements that have been published from experts and organizations. This can be fairly straightforward if your analyte has a well-established association with a clinical condition, or if it has a very long publication history. But suppose your IVD is designed to detect a novel analyte, or if there's limited evidence regarding your analyte in published literature. In this case, you may need to rethink the strategy necessary to establish your scientific validity. But this is why it's recommended that you begin this scoping process very early in your PER preparations, because limited data supporting scientific validity will significantly impact the quality of your PER as a whole. And finally, you will need to define the competitive landscape. The competitors are different than the alternatives, so keep that in mind. In the competitive landscape, this is where you will be establishing safety and performance measures, as well as your acceptance criteria, using data that has been extracted from the many articles retrieved through your competitor search. Your goal is to compare your IVD to the competitor IVDs to show that your IVD performs as intended and meets pre-established acceptance criteria. Now, be prepared to document each of these searches in detail and present your results very clearly. The process of building the SVR template relies heavily on the results of your systematic literature reviews. But don't forget that the SVR must document this, as it's a process, and the entire process must be objective, reproducible, and well documented. The notified body will expect to see that relevant articles were identified, screened, and appraised in a transparent, clearly defined manner. There are two different systematic literature reviews presented here, which we will discuss in additional detail later in this presentation. So, as we just illustrated, the SOA will be a critical component of your PER, but it's also vital to establishing the scientific validity. So what do we mean by state of the art? Typically, when we describe something as being state of the art, we're using that term to convey that something is cutting edge technology. So based on our extensive experience with MDR, MedDev Rev 4 is really our best source document for guidance as to the methodology and the required content for a state-of-the-art section within the SVR. Manufacturers are expected to demonstrate a thorough clinical evaluation of the IVD in the context of what is generally acknowledged to reflect the most current best practice science. So we need to establish scientific validity by documenting how your IVD fits into the scientific landscape, and then explain the circumstances and the diagnostic scenarios by which this IVD can be considered state-of-the-art. This means that you will be expected to present a systematic and an organized process to continuously generate, collect, analyze, and assess data pertaining to your IVD. Your goal is to develop a reproducible and an unbiased assessment of safety and performance data for your IVD in the landscape of diagnostic options and alternatives. So that's where we'll start today. What do you need to know before you can define state of the art for an IVD? We're focused on the SVR today, but remember, the goal of the PER as a whole is to effectively demonstrate scientific validity, clinical performance, and analytical performance of the IVD. The SOA is the link that connects each of these independent reports, largely by providing clarity about the purpose of the IVD, the position of the IVD within the scientific landscape, the standards by which you will measure safety and performance, and the comparators that will be used to define best practice within the scientific landscape. The presentation today is focused specifically on the state of the art, but that will require having many of these ducks in a row prior to even beginning to write your SOA. So as you're planning your SOA, you must have a firm vision about a few critical points. First, strategy. Is your regulatory strategy finalized, and have decisions been made with respect to IVD groupings or families? Are you writing one PER or multiple PERs? This is a really important decision to make early on, certainly before you begin writing your SVR. And next, your feeder documents. What is the status of all of the necessary inputs for your PER and your SVR? Presumably, you're still at the stage where you're composing many of these supporting reports, including the SVR, but do you have all of the inputs necessary to prepare those documents? Have you completed your systematic literature reviews? Do you have the data? Has your IFU been finalized? Product inserts available for review? Now, the APR will document the analytical performance, and that may include data obtained through in-house validation experiments, as well as any reports on your IVD that have been published in scientific literature. This is where you need to have a thorough understanding of the scientific standards to explain how we define sensitivity and specificity and how your IVD compares to these established standards. The CPR will present the clinical data identified through another independent systematic literature review. This includes safety data in terms of risk for incorrect test results, as well as data that presents on the performance of the device in a clinical setting. So knowledge of the patient population, indications, comparators, and outcomes, or PICO, is necessary to conduct an unbiased assessment of safety and performance. Which leads us to the SVR. The SVR must clearly present the scientific landscape by explaining the science behind the technology and ultimately what constitutes state of the art. This means you need to have an understanding of the safety and performance objectives that will be used to develop your APR and your CPR. If these objectives are not supported within the SVR, your reports may feel disjointed rather than the independent yet complementary pieces they're intended to be. Now let's discuss the systematic literature review process for acquiring this published data in order to actually support your state of the art. Additional details regarding the systematic literature review process have been discussed in great detail during our previous Criterion Edge webinars. So we will not be discussing the process in a detail. However, understanding the purpose of systematic literature review is going to be critical to discussing how to establish SOA. Thus, we should start by discussing the systematic literature review in the context of your SVR. At its core, the purpose of a systematic literature review is to ensure that you have identified all relevant scientific data using an unbiased and methodologically sound process. This is a good time to point out that this is not an optional exercise. Systematic literature review has long been considered to be the gold standard for evidence-based research, as you're all likely aware. That being said, while SLR is mandated as best practice, it can also be an extremely daunting process because this is not just simply a PubMed search. Let's refer, so as we're thinking about the different process of the systematic literature review, the first systematic literature review we discussed earlier was for the state of the art section of the SVR. This is how you're going to define the questions and identify your PICO terms, your population, indication, comparators, and outcomes. So with the help of a qualified reference librarian, you will need to design a search using proper mesh terminology and a robust search platform that allows you to search across multiple databases, from PubMed to Embase to multiple different databases that are out there. This strategy is very critical for the next stage screening because each one of those articles that you identify in your search needs to be screened for relevant data based on your defined inclusion and exclusion criteria. So if you've poorly designed your initial search, you could easily collect thousands of articles that have been retrieved, which means now you're spending a significant amount of unnecessary time and delays during that screening step. The next step then is data extraction, and that's the stage in the process in which you're going to identify the articles with specific data that's relevant to your pre-established performance criteria. In the case of many IVDs, performance criteria may encompass data such as sensitivity or specificity, false positives or false negatives, or limit of detection. And then finally, you're going to report and present the data that's been extracted, but you also need to report on the process that you undertook to actually arrive at that data. Getting back to sort of how all of these different pieces fit together, we're trying to prove the association of an analyte with a particular clinical condition. So have you given much thought to the importance and the impact of having a really solid IVDR compliant template? Many people get stuck on the PER template, but it's just as important to develop solid templates for your SVR, your APR, your CPR, and your PMS, PMCF plans. So think of your templates as your roadmap. If you sat down and read just your template for your SVR, each of these standalone reports should make sense and it should flow logically. So if you have designed an appropriate template, your PER should start at the beginning by setting the stage with the state of the art section, and the story will flow from there. The PER will culminate with your conclusion that your IVD is safe, performs as intended, and may be considered state of the art, and thus meets all IVDR requirements for safety and performance. Now we're going to take a look at the critical components within a typical SVR template and try to see how the SOA section fits in. So your template will certainly vary and should be customized based on the unique needs of your device. But in general, the approach to establishing the state of the art is essentially the same as in the MDR world. In this case, we will conduct the first systematic literature review to support the SOA section. The articles retrieved in this first search will provide the data relevant to the medical condition and your patient population. In terms of alternatives, this would include a discussion of other ways of making this clinical diagnosis, but it would also need to include a discussion of the evolution of clinical testing recommendations. Is there a specific diagnostic algorithm recommended? And this should be discussed in detail to establish how your IVD compares to alternative testing modalities. The risks and the benefits should also be presented in terms of the technology, not the patient. The clinical risks and benefits will be discussed in your CPR, but the SVR should instead focus on the potential risks and benefits in terms of this test providing accurate, sensitive, and timely results. Now, you've probably noticed that in this outline here, systematic literature review appears twice. During the PER writing process, you will be required to conduct multiple systematic literature reviews, but that also means that this entire process of defining, searching, screening, expecting, and reporting be repeated in multiple times throughout this entire process. And in fact, it's repeated at least twice within the SVR. So we're going to focus on this section in the second half of the SVR, this competitor systematic literature review, in an upcoming webinar, mainly to distinguish between the purpose and the scope of these two different systematic literature reviews. In the end, PER will help to marry the information from each of these searches together to tell a single cohesive story. But now is the time to ask yourself, does your SVR include all the IVDR required elements? A great advantage would be is if you have a template that's already been tested by having gone through a notified body review. Because IVDR is new to everyone, it's really just hypothetical whether your template will pass notified body scrutiny. Having experience with MDR and CER preparations would be extremely helpful as you develop these templates. But just ask yourself, if it flows, is it logical, and does it really tell the story of your IVD? On the flip side, though, relying on a template that is untested or if it's poorly constructed may result in downstream delays and cost overruns to fix a poorly written PER. And perhaps worst of all, it could result in costly delays in IVDR approval for devices within your portfolio. So we strongly suggest that you take the time at the beginning of your process to ensure that the systematic literature review is conducted properly and your SVR template is well organized. And that will ensure that you have established the state of the art for not only the Scientific Validity Report, but also your APR and your CPR. Some critical considerations, though: determining comprehensive PICO terms, inclusion and exclusion criteria, performance criteria, and your competitors. All of this must be made very early in the SVR planning process before you can even begin writing. So up until now, we've been talking in hypotheticals, but I thought I would illustrate this with a real world scenario. Remember, the Scientific Validity Report is showing the association of the analyte with a clinical condition. So here is a typical example of a common clinical diagnostic test, the HPV test. To give you a little background, human papillomaviruses are DNA viruses. There are 5 different genera, 48 species, and 206 distinct types of HPV. 2 of these types, numbers 16 and 18, are responsible for causing more than 70% of all cervical cancer. So suppose your IVD is an HPV test. The human papillomavirus would be the analyte that your test is designed to detect. But is it really that simple? That's a very well-established association dating back many, many years. So does that mean your SVR is just one sentence with hundreds of references? In our opinion, this is where turning to MedDev Rev 4 guidance regarding the SOA section is very helpful. The articles you retrieved in your search will provide the data relevant to the medical condition in the patient population. In this, that would be cervical cancer and genital warts. HPV is also responsible for oropharyngeal cancer in men, but that is outside the scope of your SOA because HPV testing is indicated for cervical swabs only, and therefore cannot be used to diagnose HPV infection in people without a cervix. In terms of alternatives, this could include a discussion of pap smear, but it would also need to include a discussion of the evolution of clinical testing recommendations. These have recently changed to switch from recommending primary pap smear annually to now recommending HPV testing. There is a specific diagnostic algorithm recommended as part of routine cervical cancer screening. So you want to include that and discuss how to establish HPV testing as the state of the art compared to other alternative testing modalities. You'll also want to include a discussion of risks and benefits in terms of the technology, not the patient. The clinical risks and the benefits will be discussed later in your clinical performance report. So your scientific validity report will instead focus on the potential risks and benefits in terms of this test providing accurate, sensitive, and timely results. So to summarize, you would want to describe cervical cancer and HPV and that relationship. That's going to be included in your medical condition and your background. But also consider, how does your test detect HPV? In this scenario, you're amplifying a specific gene target as illustrated in this diagram, and you're amplifying it through PCR. That is also relevant background information, and you should be prepared to justify your choice of gene targets in detail. Another aspect to consider, do all HPV tests detect the same target gene that your IVD does? Remember, there are multiple types of HPV, and each type has slight variations at the DNA level. So your SVR must therefore seek to establish that the type of HPV and the IVD that you are using is associated with cervical cancer diagnoses, and compare the detection strategy with other similar tests. If you have multiple gene targets, how does your test ensure sensitivity and specificity of your assay? Is your approach able to distinguish between these high risk and low risk HPV types? Can it distinguish number 16 versus number 33? Also consider what types of samples can be used. In this case, it's cervical swabs, but can you detect HPV in blood samples or tissue biopsies? Or what about self-collected samples as opposed to samples collected in the clinic? This will also need to be included as part of your discussion within the alternatives section. Articles retrieved during your SOA systematic literature review will help you identify whether all HPV tests work under the same premise using similar technologies, and that in turn will allow you to support your choice of competitors. And finally, you want to establish the risks of this test in terms of false positives and false negatives. Are there certain acceptable ranges for sensitivity and specificity in terms of cervical cancer diagnosis and screening? Are there benefits of HPV testing over pap smear that establish that HPV testing is a scientifically valid approach to cervical cancer screening? All of this will be included as part of your scientific validity report, but this is also why it's so critical to plan your outline to ensure that your discussion of this association is comprehensive and that it's organized well and it's also well documented. All of this background must be established before you can really truly assess the state of the art. Okay, so the articles you've retrieved in your SOA search provide that data that's relevant to those medical conditions, the patient population, and the alternatives. But there's also that second round of the systematic literature review where you are setting up your competitive landscape. We're going to be describing the competitive landscape in a separate webinar coming up. So be sure to sign up for that for additional details about this search. In that webinar, we'll help to distinguish between the purpose and the scope of the SOA search compared to the competitor search, mostly in terms of how you approach the systematic literature review, how you screen the data, and how the data is ultimately presented. The SOA systematic literature review will provide the background and the general information, but all of the data that you need to establish your acceptance criteria and to assess your safety and performance of the IVD, that's going to rely heavily on a systematic approach to the competitor's systematic literature review. The PER is going to help combine the information from these different sources, from these different sections, to provide a comprehensive, well thought out strategy. So hopefully the information that we've presented today has helped to clarify how to establish that initial section, that state-of-the-art section for your IVD in the context of scientific validity. But we also hope you recognize the importance of developing strong templates and a solid process for actually collecting and reporting the results of your systematic literature reviews.
Thank you, Sarah. We hope at this point you understand how to assess your SVR project, for unanticipated roadblocks and delays, which inputs have the greatest impact on the state of the art, how the SOA and the SVR are related, why the SLR is a critical tool in this process, what information should be included within your SVR template, and how indecision or incomplete supporting documents can negatively impact the SOA and therefore the SVR quality and timeline. So at this point, I think we're ready to get started with taking some questions. If you haven't already sent us your questions, please go ahead and start sending those in, because we're going to start taking your questions now. Sarah, I'm going to be sharing the whiteboard so we can look at some of the questions that are coming in. Let me know when you can see that.
Okay, yes, I can see that. Okay, so it looks like the first question is getting at the low risk versus high risk products, I suppose. So which components in the SVR could potentially be simplified? Honestly, really, it's the same approach. If you have a high risk versus a low risk product, you still have to show that association between the analyte and the clinical condition. You may have a slightly shorter risk assessment, but that would be affecting your CPR more so than your SVR. Your SVR is establishing the scientific validity, and that doesn't change whether your particular IVD is low risk or high risk.
Thanks, Sarah. I didn't actually get a chance to put the next question in.
Oh, no.
Doing it right now. No, you're very fast. Here's the next question. If our NGS assay reports on greater than 300 variants, does this mean we need to provide literature searches for all variants we provide info for in our report?
This is going to be one of the biggest challenges, I think. In our opinion, if you're going to report on all the different variants, I think you're going to want to focus on setting up the technology, how the technology distinguishes between those variants. You're going to want to, at least in the SVR, include an SOA search that casts a wide net so that your literature searches that you retrieve will include information and discussions about all of those variants. I don't anticipate that they will expect you to go into extreme levels of detail variant by variant, but it is expected that you're going to show mainly how you distinguish between them and sort of the clinical significance and of each of these different variants. How would that ultimately, how would the ability to detect and distinguish between these variants contribute to the fact that your test is now scientifically valid for making a particular diagnosis? Okay,
thanks, Sarah. Next question. Is the state-of-the-art search used to cover all three parts of the report, clinical, analytical, and validity, or are these separate searches?
So there will be multiple searches that you'll conduct. Think of the SOA search as just giving you the basic background section. So if you're writing a typical article to publish, this is going to be your background. You want to set up the stage, you want to define your patient population and kind of justify what the landscape looks like in terms of all the different alternatives tests that are available. But you're not going to really touch on the clinical the nuances of the clinical diagnoses yet. This is just going to be strictly the background information setting the stage. It will ultimately inform and support the information that you're going to later present in your CPR. So if there is a particular patient population that is indicated or isn't, so like in the HPV example, you would have to describe why that HPV can affect both can cause cervical cancer as well as oropharyngeal cancers. So the fact that your test is able to detect one but not the other, that would be described in more detail in your CPR. But the SOA should at least introduce that concept so that it's clear how the tests work and who they work for, and maybe some of the limitations of the tests.
Okay, thanks, Sarah. Next question. IVDR Annex 13, I believe, 1.1 requires a description of SOA in the PEP. Can you describe aspects of the state of the art that are required in the PEP versus the SVR?
Well, based on our experience with writing CERs and CEPs, really the SOA section remains largely intact and between the two of them. So you're going to utilize the same background information, the same alternatives, the same landscape, and all of that within both. So I think the difference is that the SVR will go into maybe additional details with regard to how the product is designed and sort of the overall like the indications for use. So, but a large portion of your SVR will appear as the SOA within your final PEP.
Okay, sorry, I didn't get the next one set up yet.
Oh, no.
I just need one moment. All right, oops. Here's the next question. When dealing with a short history for a new technology, how can scientific validity be augmented?
Okay, so this I think is definitely going to be an issue that a lot of us are up against. So suppose you have something like COVID testing, luckily there's a lot of information that's come out recently. So you have lots of comparators and lots of well-established relationships that that SARS-CoV-2 causes COVID-19. So when you're trying to make that establishment, you will, even though it's a very short history, there was a lot of data published in a very short amount of time. Suppose you don't have that luxury and you're not COVID-19, you're one of the other tests. In that case, you may need to justify a lot of the information that you used to basically design your test in the first place. So you may need to refer to some of your in-house experiments, some of the validation that you undertook to actually identify and characterize that analyte. And so you may not have quite as much search history to rely on, but whatever analyte you're detecting should still be within the literature. It just may not have been approved for the particular diagnostic test that you're showing. So you may have to get into a little bit more with sort of the basic science articles as part, and design your SOA search to try to capture more of those basic science articles, because you just may not have a whole long history in the clinical setting if it's a newer technology.
Okay. Next question. I know we've been asked this in the past as well. How do you approach state of the art for the different classes of devices?
So really, it's going to wind up being a pretty similar approach, whether it's a high-risk class or a low-risk class. Either way, you are still expected that you can show that that particular analyte can be used to diagnose a condition. So really, the only difference would be how much time and energy you devote to that risk section within your CPR. So you're going to clearly want to go into more detail about the risk of false positives or false negatives from a patient's perspective in your CPR. But as far as the SOA section, you're still going to take a similar approach, that the test itself may have a certain percent of false positives, or it may have a limited sensitivity in certain sample types. that approach will be the same. So here you're just establishing that this analyte can diagnose this condition. So as long as you think about it like that, you're not trying to tell the entire story, but you're trying to provide that background necessary to then understand the clinical significance when you go to your CPR, or the analytical performance. You understand why we're looking at sensitivity and specificity, why you've chosen a particular gene target or a particular analyte, and how you're detecting that analyte, sort of the pros and cons of your particular approach to detecting that analyte. So all of the nuances, really the differences in classes that you're going to be discussing will really be more reflected in your CPR and your APR, but to ensure that you are mitigating any risk of a improper diagnosis. But the SOA will be largely the same for all different classes.
All right, thanks, Sarah. Next question. This might have been asked before, but we have a few people asking something similar. State of the art is needed for the PEP, SVR, and PER. Where should we put it without repeating it 3 times?
Yeah, it will feel somewhat repetitive, just based on our CER and CEP experience. The way I'm sort of thinking about it, though, is that in the Scientific Validity Report, the state of the art is designed to tell you that association, to make that connection and to describe how that association is done. You will then want to also include some of the additional background information that's more pertaining to the clinical or the analytical performance. You want to make sure that that's tied in well within your PER and your PEP. So you may need to sort of supplement or sort of elaborate on some of the more sort of clinical or analytical sides within this section, or refer to those sections within the CPR or the APR. But for the most part, the information is going to be sort of carried over, and it should be uniform between these 3 different reports. They're supposed to be complementary. So the main contribution to the PER and the PEP that's coming out of this SVR is that background, is that association.
Okay. Next question. How should the approach differ between well-established products and products 1 to 2 years old?
I think the main way I would approach that differently is in very early on, as I'm designing my systematic literature review. If you have a very well-established product, something like influenza testing, that is going to go back decades, compared to something like COVID testing, where it's only 1 to 2 years old. So in those cases, you may want to design your systematic literature review to be more targeted, so that you will go back a longer period of time or a shorter search time. You may also need to refine some of your search terms to make sure that you're not getting a lot of off-target hits if it's a very well-established product, to make sure that you're identifying articles that are related to your specific product and not something that's similar or related or shares a very common name or a company name that could result in a lot of off-target hits. So really your main goal here is to try to show this connection that this analyte can detect and diagnose, but you may be far more limited as far as the how far back the data goes. You can rely on similar work or some of the sort of background work, the basic science work that laid the groundwork for the development of your product. That would be another approach that you could take if your product itself is only several years old, but this particular analyte was identified 10 years ago and it's only been recently developed into a clinical diagnostic tool. That you may want to go back to the basic science and explain sort of how it was characterized in that case, as providing the information you need to sort of show why it's a newer technology.
All right, we do have a few more minutes, so we're going to keep going. So if your state-of-the-art and scientific validity searches are different, how would you suggest reporting and documenting each systematic review?
So I think in this case, if you consider the state-of-the-art as one systematic literature review, and then your competitor search as a separate systematic literature review. There are different ways that you can approach that, but ultimately you're going to want to make sure you keep track of which articles were retrieved in which search. So, and you're going to want to make sure that you separate your mesh search terminology to where they're going to be searching for different aspects. Otherwise, you're going to wind up with just one giant combined search, which is not in any way going to be something that you want to try to document and report. So really, you want to make sure that you're showing, here is the state-of-the-art search. I identified 100 articles, I screened through those, and now I've selected these 10 as providing enough information about the medical condition and all of that. That's pretty straightforward. You don't really have to justify how relevant the data is or anything like that, because you're not collecting specific data points out of the state-of-the-art search. So your appraisal of the articles is based on whether or not they are peer-reviewed and whether or not they appear within a certain time frame, that sort of thing. Whereas if you're actually trying to establish the scientific validity of your competitors, there's where you have to set up your acceptance criteria and your what particular pieces of data are you searching for. If you identify randomized control trials that report on sensitivity and specificity, how do they report on that data? That's going to have to be documented separately so that you say, here are the criteria I use to actually screen and identify relevant articles. Here were my cutoffs for what I deemed to be an appropriate article for inclusion. You can't just sort of cherry pick your articles to select only the ones that sort of support your narrative. You want to make sure that it is a very reproducible, a very transparent process, particularly with that competitor search, because this is how you're establishing the landscape. What is an acceptable range of false positives and false negatives? If it looks like you may have excluded obvious articles, that will definitely get picked up on and noticed by reviewers. But really, when you're trying to think about sort of the differences between them, your state-of-the-art is trying to establish that you are within range compared to your nearest competitors. One of the analogies we like to use is sort of a sports analogy. If you're playing basketball, you wouldn't compare yourself to an NBA player, but you would compare yourself to other like 40-something women who are playing basketball. If your device performs as well as other competitors within your league, that is how you establish state of the art. But you have to know who are your competitors, who's within that league. And that's part of what needs to be sort of built into the way that you're conducting your searches to be able to make that justification. If you don't do that, you may inadvertently compare yourself to something that is way far out of your league as far as a certain sensitivity level. You wouldn't compare like RT-PCR to an antigen-based test. You know, they're detecting different analytes with very different levels of sensitivity. So you would compare RT-PCR-based tests to other RT-PCR-based tests and sort of discuss the pros and cons of which particular gene are you attempting to amplify from one test to the next. So if you keep that in mind as you're sort of documenting each of those systematic reviews and trying to understand sort of the level of detail in which you're going to need to screen these articles, I think that's sort of the best approach to take.
Thanks, Sarah. Here's the next question on the screen. How do you approach state-of-the-art for general use IVDs that are not specific to a particular analyte?
Oh, okay. So in this case here, I think you would have to basically describe the use of your IVD in a very general sense. I'm not sure about the specifics. A lot of this will depend on, like I said, each template will be slightly different based on the specific needs of your IVD. But I'm thinking maybe if your IVD is designed to detect like maybe pH, you know, you would have to use the hydrogen ion concentration. If you're trying to detect DNA, it may not detect a particular DNA sequence, but if it detects DNA in general, then you would discuss sort of how the technology works. So I think sort of framing it around the goal of the technology, and sort of alternative ways of also what are the alternatives if you were not using your IVD? What options would you have available to you? That I think that would be the best way to frame it. So you just would not have as much discussion about a specific analyte. You would sort of talk about it more in terms of the general class of analytes that are being detected.
Okay. Our next question. Which, if any, society guidelines do you reference, and which, if any, public adverse event reporting databases? Not sure if you have insight into this.
Oh, yeah. So in terms of the public adverse reporting databases, you really would not need to include any of those within the SVR. That would definitely encompass part of your PMPF and your clinical performance report. But within the SVR, you really wouldn't need to refer to those. You're more focused on the competitive landscape that have been reported within published literature. As far as society guidelines, that really varies depending on the IVD. So I generally start with whoever the clinicians would be who are using your particular test, who's actually benefiting from the information that the diagnostic information that your test is designed to detect. So if you are doing something with cancer biomarker, you may want to consult the American Cancer Society or Society of Gastroenterologists, or something like that. So it really does vary. A lot of it depends on sort of what your analyte is designed against.
Okay, we just have a couple more questions. I'll go through those. When dealing with a general use reagent, such as a DNA extraction product, the clinical condition can vary significantly. Would you recommend focusing more on the quality requirements?
Yes, I think that would be the approach I would take. So if you, just on my own experience, if I'm doing something like a QIAGEN kit versus one of the other kits that you can pick up off the shelf, you would basically try to explain sort of the technology behind it, how quality is maintained, any potential limitations in terms of the sample that's being collected, whether that's temperature or pH or the amount of DNA, the level of sensitivity or specificity. So in that case, your analyte, if you're just using something that's going to extract DNA to make a diagnosis using that DNA, your analyte is going to be the DNA in general. You just want to show that you are providing a clean DNA prep in the end. So that would be the approach I would take to that. It's not so focused on the actual DNA sequence itself, but it's a clean molecule that can then be used for downstream testing, whatever that may be.
Okay. All right. How do you define the state of the art for a new device?
So this can definitely be challenging. Like, let's suppose there's just nothing like yours out there. In that case, you have to sort of go back to the background material and identify how would that particular disease have been diagnosed prior to your IVD coming on the market. So if you're looking for something like basically prostate cancer diagnoses prior to PSA testing being introduced, how were those diagnoses made before your test was developed? And then if you happen to be the only PSA test on the market, you would still need to show that this is a marker, this is an analyte that allows you to detect prostate cancer accurately and with a high level of sensitivity that's beyond what any of the other alternatives provide. If hopefully there are other similar tests that also detect that particular analyte, you could look at something like detecting the RNA versus the protein, or look at the messenger RNA versus the DNA sequence. There may be some sort of different approaches that you could take if you are sort of standing alone in your particular competitive landscape, if there just aren't a lot of really good comparators out there.
Okay, we have sort of a broad question next. What are the trends of the medical device industry for the definition of the state of the art?
I think they've been fairly consistent, really. And that's where, referring back to that MedDev Rev 4, it really does seem like we're sort of set on this definition of the fact that you should be able to detect something with a certain level of accuracy. You know, you have to be able to show that your device performs as intended, and it meets the requirements as set forth by all of the other closest competitors. So really the definition, we're interpreting it based on our IVDR guidance as being fairly similar to what we already obtain from the medical device world. I'm not sure if that fully answered the question or not. It's kind of a very nuanced or very challenging question, but I think in general, you want to show how it fits and that it reflects the best practice science, and that other people acknowledge that your method of detection reflects a state-of-the-art technology.
Okay, this is sort of a different question. Are there differences, but also slightly broad, are there differences between the state of the art in a CER for medical devices and a PER for IVD? I know that you've done a state of the art webinar in the past, Sarah, so I wanted to remind this particular attendee that they can also kind of visit that webinar and watch that recording to get a little bit more information, but I'll give it over to you to give a short response here.
So as we've been developing templates for some of these different IVDs, one of the main things we're noticing is just how do we define you have to not only define the medical condition, but you also have to define the diagnostic option in terms of the analyte itself. Whereas in most of the time in the SOA for a CER, you're going to set up how this particular device performs and how it's used to treat a medical condition. You're not really getting into the diagnostic side of things. So you're kind of switching gears from focusing in an SOA in the CER, you're focused on how that device is used to treat a particular condition. Whereas the SOA in a PER is focused more on how that particular test is used to diagnose and inform clinical decision-making. So I think that would be the main difference. A lot of the sections and a lot of the templating will be very similar, but you will want to focus more in on the diagnostic criteria and the analyte choice.
Okay, we just have 2 more minutes left, so I'll squeeze in just maybe one more question. When building a search for SVR, it seems it would be a combination of Analyte with the condition. Should it include methods, alternatives also?
So yes, definitely. You're going to want to include a discussion of alternative technologies for detecting that particular condition. So this gets into sort of the importance of really thinking out your strategy as you're designing this SLR, or it can get really ugly looking very quickly. If you don't have the proper mesh terms, you may retrieve thousands of off-target articles and hits. So, and you'll be expected to screen all of those. So if you're trying to detect any articles that discuss your analyte with regards to this particular clinical condition, it's going to require some sort of tweaking of your search terms and your search strategy to really ensure that your search is targeted yet broad enough that you're not excluding any really obvious articles. So you may have to play, it's never going to be the very first search you try. It's very rarely the final search strategy. You usually try to employ several different changes to the terminology and then do sort of a quick screen to make sure that you're really capturing all of the different articles that you should be retrieving. So you want to go in, and that's why it's so important to document this whole process so that in the end, you can justify any decisions that you made to include one search term or exclude one search term. That will all be included within sort of your discussion of the search strategy.
Okay, Sarah, I know I said that was the last one, but I did want to squeeze in one more question. What is MedDev Rev 4? I think this is a pretty important one. And what is the full reference, and what does it have to do with IVDR?
Yeah, so we refer to this quite a bit in the CER world. It's sort of one of these intuitive, but this is a regulatory document that provides a lot of details about sort of what they expect in the medical device world in terms of what are the reporting requirements, sort of what information should you be expected to include within your CER. So we refer back to that for understanding what an SOA is. It gives a lot of really good details about sort of how the SOA is structured. If you search for MedDev Rev 4, that particular source document should pop up pretty quickly. It's a very common source document, but it will provide a lot of helpful insights as to sort of what they mean by state of the art and how they expect you to document state of the art.
Wow, thank you so much, Sarah, for answering all those questions. We definitely took a lot, and we have a couple more that we didn't quite get to. So if we didn't get to your questions, we'll reach out to you afterwards. Thanks, Sarah, for hanging on for a little bit later. So thank you again, everyone. Thanks to all our attendees. Thank you, Sarah, and everyone for joining. So take care. Have a great day.